started, I want to mention to the audience that you can submit questions via the web portal or to me on email at tyler.vanburen@cowen.com. With that said, let's go ahead and start with the B-cell lymphoma franchise, naturally with FT516 and 596. So CD16 is the backbone of Fate's INKs and core to your strategy in terms of enhancing ADCC. But the question I wanted to ask is, do you believe many are still kind of missing this point by grouping you into the allogeneic CAR T bucket?
Yes and no. I mean, I think NK cells are different than T cells, right? So I do think that NK cells have different mechanisms of action, obviously, that can be exploited, that can add value to lymphoma patients. That said, T cells have obviously cell-based therapies with T cells. They're a proven modality. They're quite successful for patients. And ultimately, I think when you think about competing in the marketplace, yeah, we have to recognize that, NK, any therapeutic modality is going to compete, right? So we have to think about the development of an NK cell therapy in the context of there are absolutely successful CAR T cell therapies. And I think we have to recognize that. And I think given that, again, we should recognize how NK cells may be different than T cells and what are the unique mechanisms associated with NK cells that we can exploit.
Well, on that topic, as we look 5 to 10 years from now, how do you view the addressable patient population for INKs relative to CAR Ts?
I do think potentially, and again, let's give ourselves some time to play it out, but I do think potentially NK cells have a much broader patient reach for several different reasons. So far, and again, it's an evolving landscape, but so far NK cells have shown, I would say, a pretty favorable and differentiated safety profile compared to T cell modalities. And I'll actually include, for instance, engagers as well as T cell therapies in that bucket. I do think that when you start talking about moving earlier line and stepping into, for instance, a community setting and trying to plug into well-established regimens that are commonly used every day to treat patients in early line, I think safety is important and safety matters a great deal. And so I do think that because of that, the patient reach of an NK cell may be broader than a T cell based modality. In addition, back to sort of your first question, which I think is a great question, I think the patient reach can also be advantaged with an NK cell because of NK cells' mechanisms of action that you may be able to exploit. Back to the CD16 receptor, CD16 allows an NK cell to synergize with a monoclonal antibody. And that's one of the main mechanisms of action of an NK cell, which is highly differentiated than, for instance, how a T cell will behave. And I do think, obviously, whether you're talking about lymphoma, myeloma, many solid tumors, monoclonal antibodies that synergize with NK cells are used early and often in treating patients. And if we can plug into those regimens, again, I think that speaks to the benefit and the breadth of potential NK cell therapy.
Sure. So getting into the specific programs, you guys are pursuing franchises with iterative development programs. And on the recent earnings call, you said that you're deciding between 516 and 596 for the pivotal program. So what are your latest thoughts on consolidating or better defining the B-cell lymphoma franchise strategy for the pivotal that should begin in the second half?
Yeah, so I mean, I've always maintained that I believe the idea of multi-antigen targeting is and will be best-in-class approaches for therapeutic regimens to treat patients, right? The idea that you can attack a tumor from multiple different angles, I think for us has always been very core to our belief system on how to drive best responses for patients. And so for that reason, obviously, we've characterized 596 as a best-in-class product candidate. We built 596 off of 516. 516, obviously is more advanced today relative to 596. We've seen, I would say, very encouraging data with 516, but we're working hard to have 596 catch up, if you will, to where 516 is today. And we hope to, and we're essentially marching through the year almost in an agnostic way as to, whether or not we can conduct that study with 516 or 596. I'm not going to apologize if 596 catches up and replaces 516. It was almost always the intent in building 596. We were building off of 516. It was a next generation approach. And we do think it's a best-in-class approach with multi-antigen targeting. In addition, as you know, it has the IL-15 receptor fusion. 596 has the IL-15 receptor fusion engineered into the cells. And we think potentiating the cells with cytokine support engineered in, will improve activity, as opposed to relying on Cy/Flu or exogenous cytokines to potentiate the cells.
Okay. And when you look at the ASH data, obviously it's still relatively early in terms of comparing 596 with 516, but the combination actually, I'm getting into the combination of Rituxan now. When you look at the combination regimen of the CR rate, it looked improved versus the monotherapy. So why not just prioritize the combination moving forward?
With 596?
Yeah, with the combination of 596 and Rituxan.
To be clear, we are. So when we think about using 596, again, back to the design behind the molecule itself. The molecule itself was always designed to enable dual antigen targeting. And obviously you exploit dual antigen targeting by combining with rituximab on top of the CAR-19. And so as we think about it now advancing development, thinking about obviously expansion arms, and then ultimately a pivotal, the strategy would be to completely exploit that dual antigen targeting nature and combine with rituximab.
Okay.
Yeah.
Will pivotal trial focus exclusively on large B-cell lymphoma patients? And, obviously there's been a mix of patients enrolled previously. Will it be easier to enroll those patients now that you guys have the data that was reported at ASH?
Yeah, I think, obviously we're gonna have the conversation. We have RMAT designation for FT516 at this point in time. We will likely file for RMAT designation for 596. That's part of the conversation we're having with the FDA, plan to have with the FDA in the first half of this year, what that first pivotal study looks like. We've talked about the idea that we believe that pivotal study will be in a patient population that is downline of CAR T-cell therapy. We think that's a terrific opportunity, a potential fast to market strategy. Obviously, as sort of we alluded to, the CAR T-cell market is expanding. And if that market is expanding, there's very likely going to be more patients downline of CAR T-cell therapy. So we think it's a great place to go, especially in exploiting the CD16 receptor in the combination with rituximab. Whether or not it's harder or easier to enroll, I think there are quite a few patients that are progressing or failing on CAR-19 T-cell therapy. It's only curative for probably 20 to 25% of patients.
Okay.
Which means obviously 70 to 80% of patients are available for treatment.
Yeah. So regarding the dose escalation for 596, you're moving to a 2-dose schedule on day 1 and 15 with up to 2 cycles. So how do you determine who gets the second cycle, and how well tolerated is a second dose of Flu/Cy?
Yeah, so I'll take the second part of that first. Keep in mind the FT516 treatment paradigm was always 2 cycles with Cy/Flu in each cycle, right? So I think we could probably agree that the FT516 safety profile, which included 2 cycles of Cy/Flu, is pretty favorable. And so, yes, I think that the 2-cycle regimen, if you will, is very well tolerated in patients. How patients advance to a second cycle is basically off an assessment at day 30 with respect to tolerability and clinical benefit. So if the patient is tolerating the therapy and there's evidence of clinical benefit, a patient is now eligible for the second cycle.
Okay. Do you have enough data now to say that second cycle is clearly adding to improved response rate or depth of response, or is it something that we might see have a more significant impact in terms of durability response?
Yeah, so this is a great question, and I think we're still assessing this at some level, and the reason for this is a couple-fold. I believe that the second cycle can add clinical benefit. So we certainly have patients that have gone from PR to CR at the end of the first cycle versus the end of the second cycle. So the second cycle, has continued to convert patients into deeper responses. We've actually had a patient on 596 that was stable disease that went to CR with the second cycle. So I do think the second cycle can add benefit. All that said, the vast majority of our patients that achieve a CR do achieve it at the end of the first cycle, whether that be with 516 or 596. And that sort of speaks to the point that I do think, again, not that additional cycles can't add benefit, but these cellular therapies, including patient-derived CAR T-cell therapy, they're very potent, they're fast-acting, and I think, part of the therapeutic objective, if you will, in delivering these cell therapies is to deliver a knockout punch, if you will, within a relatively short time frame to lead to best response and durability of response. Mm-hmm. And I think that's true of patient-derived CAR T-cell, and I'm not going to be surprised if that's true of 516 and 596.
Yeah. Do you think that second cycle becomes more impactful in like a more indolent disease like myeloma or more difficult to treat tumors like solid tumors?
Potentially, yeah. I mean, I do think as you get into tumors that are more difficult to treat, and this would include solid tumors, obviously, that second cycle and beyond can certainly be important. Yeah.
Got it. Okay.
But I do think, and again, we'll get some data with myeloma and we'll look at sort of the patient-derived CAR T-cell experiences with myeloma. But I think in lymphoma, and this is going back to like the early days of Kite and Juno, I think that's pretty similar to what they saw, that, during that first window of sort of 20 to 30 days, that sort of knockout punch proved to be pretty critical to long-term durability of response.
Yeah. And then the 596 study schema says that patients are eligible for retreatment following disease progression. So assuming that would be after that second cycle in most patients. And so maybe you could clarify that. And how do you determine if someone's a candidate for treatment after progression?
Yeah. So this was not always on our protocols. Our protocols stopped it, for instance, 2 cycles. We now have the ability under our protocols for 2 different dynamics to occur. So let's say a patient gets to the end of the second cycle and they're in a response. Depending on how that patient is doing and patient and PI interest, we actually do have the ability to deliver a 3rd and 4th cycle. So we have started looking at, depending on how the patient is doing and if they're responding and if they're tolerating therapy well, we have had patients and physicians and patients and say, hey, let's give a 3rd cycle. So that is starting to look at that at more than 2 cycles with patients in response. We now also have opportunity under our protocols where a patient at the end of the second cycle who has responded, and then let's say they run out 8 months and begin to relapse, we now have the opportunity to re-intervene. And again, that is a patient-physician decision as to whether or not that patient qualifies for the study and would like to participate in the study.
Yeah. And that would require another round of flu-cy, right?
So let's say, they're down the road 8 months. Yeah, that would require another round of cycle at that point in time. Yes.
Yeah. Have you guys treated, post-second cycle, retreated a lot of patients, or is this something?
I wouldn't say I would not say that that's sort of the standard paradigm. Our protocols do allow for it, and it has happened.
Okay. So
I wouldn't put it as the standard paradigm.
Understood. Okay. That's clear. How many patients are now being treated inpatient versus outpatient across the various studies and cohorts?
With our NK cell products, give me a little latitude here, but almost I would say probably 80% plus, at least, are totally outpatient.
Okay. Won't quote you on that, but obviously
it's high. I think with 516, I think we disclosed at ASH, and I think at 596 we disclosed at ASH that it was very high. It was about 80% of patients were being treated outpatient with the product candidates. Yeah.
All right. I wanted to ask you about the 516 Benda and 596 RCHOP cohorts. What portion of patients in these trials are being treated in the community setting, and when should we expect data updates from these cohorts?
Yeah, so the 516R Benda expansion is open for enrollment, right? So that currently is open for enrollment, and we're enrolling patients and generating data. And certainly we'll look to the second half of this year potentially to give an update on the 516R Benda experience. Those are patients, as we sort of just alluded to, can be either treated outpatient or more in a community setting. And our goal, and in continuing to activate sites, is to activate sites that are more in the community. So we are reaching into the community with our site activation strategy. With FT596, as you have alluded to, we are looking to go very early line. We're looking to plug into a standard RCHOP regimen in first and second line patients that will absolutely take us into the community setting. That study is not yet open. We're submitting that protocol to the FDA and hope to open that study in the middle of the year.
Okay. Let's move to myeloma and AML. The data for both of those indications are now expected later in the year in the second half. So it's maybe a bit later than some might have thought. So can you give us an update on the status of these programs and the progress that has been made?
Yeah. So FT538, let's talk about AML first, right? FT538 is in dose escalation in the relapse refractory AML setting. Most of these patients are multiply relapse refractory. We are now at the 1 billion cell dose level with FT538. We can continue to escalate under the existing protocol to actually 1.5 billion cells per dose. And our intent is to complete dose escalation with FT538 through 1.5 billion, and then provide an update on the AML franchise, which would include a comparison of the first generation product 516 versus the second generation product FT538. And so I hope to be in a position to do that in the second half of this year based on currently where we are in dose escalation with 538 in AML. In multiple myeloma, 538 is also being evaluated in multiple myeloma in combination with daratumumab, which we think is super exciting. And now 576, which is dual antigen targeted CAR BCMA, delivers as a monotherapy as well as in combination with daratumumab. We've now started enrolling patients with 576 as well. So I don't think we'll necessarily complete dose escalation with 538 and 576 in myeloma, but certainly looking to give an update. I think in the BCMA targeting space, there's been some pretty disparate responses with respect to BCMA targeted therapies, whether we're talking about the cell therapies or even the engagers. And we are using a novel binding domain for BCMA. So, I think, showing proof of concept that our binding domain is working and we're seeing sort of responses with that and sharing that data set, I think is an important data set to at least provide investors an indication that, hey, our BCMA binding domain has activity, has the potential to have activity. Because unlike on the CD19 side, I think we've seen some pretty disparate sort of responses with respect to BCMA targeted therapies.
Can you talk a little bit more about that binding domain? Obviously, Beckmaz is different from Carvykti, and Carvykti is using the VHH binder, and some people have said that might be leading to their differential efficacy, but curious to hear more about your guys' novel approach.
Yeah, I mean, we in-licensed the binder probably about 3 years ago now, maybe a little over 3 years ago now. We've been optimizing it over the course of time. I think one of the reasons we're excited about the binder is in preclinical models of sort of, stress and low levels of expression of BCMA, we've seen activity. So at least preclinically. So, that was one of the things that we felt differentiated that binder. BCMA expression can vary significantly on myeloma. BCMA can actually be shed and can cause some challenges with shedding. And so one of the factors that we liked with respect to our particular binder preclinically, and we thought it was differentiating, is we saw activity in myeloma and in preclinical models where the density of expression was very low.
Interesting. Last question I had on myeloma is just the combination with Dara. Are you seeing preclinically, or based upon the data you have so far, are you seeing similar synergy to what you see with Rituxan and 516, 596? Or
yeah, absolutely. I mean, this is one of the things that we were drawn to very early on, back to the concept of multiple antigen targeting, and we always felt like we could build differentiated products based on multiple antigen targeting and plugging into the monoclonal antibodies that are used earlier in care through the CD16 receptor. Yes, and so 576, it's a very similar dynamic with 576 that we see with 596. That if you test, for instance, gosh, what is the activity through CD16? You get X. If you see, gosh, what is it through just the CAR? You get Y. And if you go with both and you exploit both, you see pretty profound synergistic activity.
Okay, great. Let's move to FT819, your track-targeted CAR-T or your IT. When should we expect more data from this program?
So we haven't disclosed any clinical data yet, and I think we are planning to provide an update on 819 this year. Again, I don't think we'll necessarily wait to get through dose escalation all the way. This is the first ever, I believe, iPS-derived T cell, not even CAR T cell, first ever iPS-derived T cell in the world to go into a patient. I think there's been a lot of speculation, not on our part, but out there in the world, about whether or not you can actually make a functional alpha and potent alpha beta T cell from an iPSC. A lot of speculation on that. So, we're excited about the 819 program and look forward to sharing a first clinical update on 819 sometime this year. I think, preclinically, we gave an update at ASH, both with respect to the data that we're seeing and benchmarking against patient-derived, I shouldn't say patient-derived because it's preclinical, essentially primary T cells against iPS-derived T cells. And we feel really confident in the data that we're seeing preclinically. And we also provided an update on our manufacturing paradigm and a little bit of the profile of 819 and what it looks like going into patients. And again, a pretty high component of the product is of the sort of memory phenotype. So, again, it's been a long journey, but I feel like, we've gotten to the point where we think we're making a really potent alpha beta T cell product.
So I was looking back at the study design, and it has the 3 regimens, A1, which are pretty straightforward, and then B, where you administer 3 CAR IT doses on day 1, 3, and 5. So can you, help me understand why you guys decided to explore that regimen?
Yeah, this sort of goes back to a little bit of what we were saying. I mean, I'm super excited about T cells, right? And the potency of T cells. How could you not be, based on what's been shown in both lymphoma and myeloma? They tend to come with toxicity, right, at some level, especially when you want to compare that to an NK cell. And that's true of both, I think, T cell engagers and CAR T cell therapy. So there's been some exploration certainly in the engager world, but also in the cell therapy world, about what's called sort of fractionating dosing. And so that was one of the things we wanted to explore in this phase 1 study. Could potentially, with a multi-dose regimen, could you drive potency and potentially reduce toxicity through a multi-dose regimen? And so there's been some work, certainly work done in the engager world as well as the cell therapy world with T cell modalities to try and reduce toxicity by, what's called fractionating dosing. And that was essentially the one of the arms that we wanted to explore.
Okay, very interesting.
Yep.
So solid tumors, can we expect an update later in the year, maybe at SITC? And just what should we expect from that update in terms of FT538 in combination with standard care antibodies?
Yeah, it's FT538. It's in four different FT538's in four different arms, right, in combination with monoclonal antibody. Certainly, looking to give an update in the second half of the year. SITC looks like a reasonable place to provide that update. Again, this is multi-dose, multi-cycle regimens in combination with monoclonal antibody. In addition, as you know, we cleared an IND in January with the first iPS-derived CAR NK cell product, FT536, for solid tumors, and looking to dose the first patient with FT536 in May or June. A super novel CAR associated with FT536, really interesting and fascinating biology, including how we may be able to overcome one of the main mechanisms of tumor evasion in solid tumors. And so, again, back to novel CAR, interesting biology. Sure, would certainly like to give a first update on what we're seeing. It'll be early, but what are we seeing in sort of initial clinical experience?
Okay. A lot of different FT numbers to track there, but FT538 with the antibody combinations, is there one antibody combination in solid tumors that you're most excited about, or do you truly just have to wait and see the data?
I think we have to wait and see the data. I think if you're looking to exploit ADCC and leverage CD16, I think some of the strongest data that's out there is with cetuximab and trastuzumab. Suggesting that they are certainly ADCC competent and can be exploited for ADCC benefit. I think that's been at least shown in clinical studies with respect to cetuximab and trastuzumab in clinical studies. So I think that they're two interesting combinations. I do think the combination with checkpoint inhibitor therapy generally is super interesting as well. The idea that you can go down line of checkpoint inhibitor therapy, use an NK cell to essentially rejuvenate a T cell response and bring together innate and adaptive immunity. I think, while that might be more conceptual right now, certainly we've demonstrated the value of that preclinically, and I think clinically that can make that can be pretty powerful mechanism of action, bringing together innate and adaptive immunity and essentially rescuing patients where their tumor has evolved to evade T cell recognition.
Interesting.
And that goes back to sort of the beginning. How do you think about exploiting an NK cell versus a T cell? And I do think, people want to sort of conflate, it's a cell therapy. I think you got to think about, them very differently. An NK cell is very different than a T cell, and how you want to deploy an NK cell versus a T cell. There's lots of unique opportunity for both.
Got it. Okay. We're up on time here, but to wrap up, I'll ask you the question that I always like to ask at the end of the fireside chats. Scott, what aspect of the Fate story do you believe is most underappreciated by investors?
Interestingly enough, believe it or not, I think people bucket Fate today as an NK cell company. So I do not think there's much appreciation of the fact that Fate is developing off-the-shelf T cell therapy, and that sort of what we were just alluding to. NK cells and T cells have unique and differentiated opportunities. And ultimately, I do believe they can be used together. And I believe Fate is really the only company that can bring together off-the-shelf NK cell and T cell therapy.
That's great. Great way to wrap up. Scott, thank you very much for your time. Enjoyed the discussion. Thanks, everyone, for logging in.
Thank you so much.